So plasmids are going to have an important role in this course, and we need to be certainly familiar with them. So just a few things about their terminology. When we say plasmid, we're talking about a relatively small circular DNA molecule that's present in bacteria. And these can be, on the order from a couple thousand to tens of thousands of base pairs. And when we use the term recombinant DNA, we're saying that we're assembling DNA from more than one source. So we're using laboratory methods. We're taking E. coli DNA, maybe a gene from a virus. Maybe we're putting a human gene in there as well. So recombinant, multiple sources. Now the word cloning, in a very general sense, means that we're producing genetically identical organisms. We say molecular cloning. This is when we're using several of these methods to assemble recombinant DNA molecules and to replicate those, amplify those. In other words, clone the molecules. But we often say the word cloning when really we mean this phrase down here, molecular cloning. So when I say cloning, I'm not talking about sheep. I'm talking about plasmids in DNA. And so a cloning vector is a recombinant plasmid that could potentially contain DNA from E. coli and viruses and other things like that. It's double-stranded, and it's usually in the order of 2 to 10 kilobase pairs, depending on the particular vector. So there are different types. There are things we call shuttle vectors or just simple cloning vectors that are used to make many copies of a gene if you want to amplify a gene. What we're more interested in, however, are vectors that enable the production of protein, called expression vectors. In other words, protein expression vectors. And we're going to use one called PET28. This expression vector, we have a map shown over here. This contains a few important features. The first of which is called the multi-cloning region, or the MCR. And that's actually kind of what we have right here. Sometimes called the MCS. This has a lot of restriction sites. We'll talk more about restriction enzymes in a little bit. And this enables us to insert the desired gene in that particular location. This also contains antibiotic resistance gene, kanamycin, or CAN-R. This is what we would consider a selection marker. It means that we can give this plasmid to bacteria and then use antibiotic kanamycin to kill off all the bacteria that did not take up this plasmid. And so we can then be sure that any bacteria surviving contain this plasmid and therefore contain whatever gene we put into that MCR. This also has an ORI, or an origin of replication. This enables the DNA to be replicated, the plasmid to be replicated, perhaps very, very many times. A lot of times the goal is amplification, and so the origin of replication ensures that very many copies of this plasmid get made when it's inside of a bacterium. There are also elements from the lac operon, not the entire lac operon. It does enable protein expression to be inducible, and that's sort of what this lac I is here. We're not going to get into too much about that right now. There's a video where we'll talk much more about that. But for now, just understand that there are elements on here that enable us to turn on protein production. And what we're going to do first with plasmid is purify it. And this is going to be a plasmid that's grown and amplified in E. coli, our PET28 plasmid. The cells are going to be lysed under high pH or alkaline conditions. A lot of times we call this alkaline lysis mini prep procedure. So the alkaline conditions certainly break open the cells. Sodium dodecyl sulfate, that SDS, will enable the proteins that are in there to be completely denatured. We also add RNase, which is an enzyme that breaks up RNA, and there's a lot of RNAs from the ribosome to messenger RNAs present in E. coli cells, so those RNAs need to be broken up. The solution then is neutralized, and that causes a lot of the cellular debris, things like genomic DNA, many of the proteins, cell membranes, a lot of those things will precipitate. And then we can centrifuge that and collect the supernatant. And the supernatant is going to contain the DNA, the plasmid DNA we want. It's not going to contain the genomic DNA because that's been precipitated. It's going to contain some proteins as well. And so we have to then have another purification step. And so we apply that supernatant to a spin column shown here. So we'll put the DNA solution, or the supernatant solution, into that spin column. and it contains a silica. This disc right here that you can sort of see is actually a silica disc, and the DNA will definitely stick to that. Think of all those phosphate groups. DNA and silica will have an interaction. And it's washed with ethanol. Now the ethanol is going to wash away the contaminating proteins. Proteins interact well with ethanol because ethanol has this sort of polar yet also nonpolar components, just like proteins do. But DNA is very, very polar, especially double-stranded plasmid DNA, and so it's definitely not going to wash away in the ethanol. It's going to stay bound to that silica, and so that's going to enable us to wash away those contaminating proteins, and then we're going to use water to elute the DNA from that spin column.